Hi, my name's Morris, and welcome to this NeuroCave Plus demo. I'll be guiding you through an exploration of a Allen Brain Institute donor's brain data set, which appears in the foreground here. And this data set is a neurogenomics data set, so it's from the Allen Brain Institute, so it has an MRI component where there were 8 donors and 4 of them have DTI imaging modality on their MRI, and so you can see the raw image data there. They also have microarray data accompanying in this data set. So they had 62,000 probes per microarray, and they did 500 samples per hemisphere, so it's almost 1,000 samples per brain, and they mapped out the samples with their 3D anatomical coordinates, and that's what you were seeing there were those coordinates. And they allow you to search for genes, so if I'm going to look up my favorite monoamine transporter, it'll come up here with the little heat map that you can roll your mouse over, the red and green hot and cold patches, and it'll show you what there looks like. SLC18A1 is the gene symbol for that. That's SLC. Let's see what all the SLC18s are. Looks like there's a family of 4 of them. That's the solucarrier family, vesicular monoamine. So you can see these hot and cold patches in different brain areas. And so NeuroCave is a bit of a more interesting, unique way of browsing the data sets like these. So you have the anatomical positions of all the sample sites over here on the left window, and that's in the foreground. In the background there is a UMAP embedding of the gene set, and you can adjust the sizes of the glyphs independently from each other. So what you're seeing on the left is the foreground anatomical, and the background, the distance, the genes, and on the right you have the gene data set in the foreground, and there's that little brain in the background there. When you explore this data set, you can do a search. We can do a search for SLC18. Well, that one's not there. Let's see if there's any other SLCs. Yeah, there's an SLC. There's another SLC. There's a few of them. A lot of these. So you can see the connections of the genes to other genes. Well, those are co-expression networks, and these connections, edges going to the brain representation over there, they represent brain regions that express the genes that we selected, and you can see them in this view extending from the gene set UMAP embedding in the distance over to the brain regions in the foreground. You can adjust the opacity of the edges and turn on some animations to see exactly which brain regions and which genes are active and selected. We can adjust the gene to gene edges and the gene to brain edges, the edge weight thresholds, to show more or less genes or gene connections, intragroup and intergroup. We can also switch the threshold mode to show the 12 highest weighted connections per node, and we can control which grouping we're interested in. So we just turned off the intergroup edges, so now only the intragroup edges are visible. We can turn off the skybox if that's distracting. We can unlock the color codings for the left and right windows, so we can keep the anatomical coloring on the left, and we can select the long non-coding RNAs coloring. So I pre-filtered the pre-annotated the data set with whether the protein or whether the gene codes for a protein or encodes a long non-coding RNA, so we can make the protein coding genes translucent and just show the long non-coding RNAs, so we can click on them and see what other genes are co-expressed with them and bring out that network. We can also search for brain regions, so we can search for front. So that's cingulate frontal part. We get a couple, so we can see its edges. We can click on some of its neighbors and kind of flesh out a network, and then maybe can turn the intergroup edges back on, and we can see which genes are expressed in that network. Opacity down so you can see through those. So that's how this data set can be explored. There are also different coordinate systems, so we can switch from the UMAP coordinate system to a platonic solid based coordinate system. Change, so we can see how the brain are the multicolored spheres, and the two colored blue and pink cubes are going to be the long non-coding RNAs and the regular protein coding RNAs. We can change the color coding back to the WGCNA for the module colors. We can really see which modules are expressed in our frontal network and connected to the long non-coding RNAs, which is all the edges we enabled. So that's how I'm envisioning that the neurogenomics data sets from Allen's microarray and MRI brains can be explored. Thanks for watching.